Welcome, life science learners, to another installment for you guys. Today we're focusing on cells. It's an important section. Trying to understand what a cell is crucial to understanding the processes in life sciences. Fundamentally, a cell is where life starts with. So we've got to look at what a cell is, the components of a cell. Let's try and understand what makes up a cell and look at functioning of the different parts in a cell. So, in our lesson today, we're going to focus on various concepts. Let's try and unpack a question first. Have you ever thought about what is the smallest unit of life? So, what's the smallest living thing that we've come across? I mean, we've heard of viruses, we've heard of bacteria, we've heard of things that are microscopic. And these are often things that have the ability to survive individually as a single unit. And often these are the smallest living organisms that are unicellular, made up of a single cell. So we've got to understand that a cell is the smallest unit of life. As an overview of our lesson, let's look at what we're going to be doing in this lesson. So we're going to talk about the cell theory and the cell as a unit of life. We're going to broadly classify cells based on their structure. We're going to spend some time looking at cell size and trying to understand what the size of cells are in relation to other organisms. We're going to spend some time looking at animal cells, their structure and their function, and we're going to compare that to parts of a plant cell, and then we're going to differentiate the concepts and the structures between a plant and animal cell. Let's get straight into some key words during this lesson. I might pause and elaborate on these, as there may be some concepts that we need to unpack as we get through them. The first keyword is a cell, and we'll refer to what the cell is when we discuss the characteristics of a cell and the theory of a cell. We know that a cell has a membrane that encloses its internal contents. We're going to look at the structure of a centriole as being an organelle, a smaller part in a cell. We're going to look at centrosomes as being structures. A very important part is the chromatin network, so the thread-like structures found inside the nucleus, which we refer to as the blueprint of the cell. We're going to look at those. The cytoplasm, so the liquid part within the cell membrane. So the cytoplasm, the internal contents of the cell. We also need to look at the endoplasmic reticulum, so this maze of folds of connective structures that connect the nucleus to the rest of the cell. Within the cell, the endoplasmic reticulum. We also need to look at structures called the Golgi body, and we will look at the structure of this. The mitochondrion as an important organelle which plays a significant role in cellular respiration. One of my favorite organelles called the nucleus, which we often refer to as the control center of the cell, and how that membrane encloses the contents of the nucleus. These are structures that make up a cell, and they're called organelles. We're going to look at a concept called phagocytosis. Basically, it's how a cell will engulf, or an enzyme can be used to break down certain substances, so that's phagocytosis. And structures called pseudopodia. So pseudo meaning false, and podia referring to feet. So structures that are used to move organelles or cells. An important concept or organelle is the ribosome, which we need to remember is crucial to protein synthesis. A vacuole, which is an organelle that has a specific function in animal cells, but very important in plant cells, which makes up a large component of the cytoplasm. Important to plant cells are an organelle called the chloroplast, which we often refer to as an important part for photosynthesis. The shape of cells can be defined or described as being flaccid. So when a cell has lost its shape, it's flaccid, or when a cell has maintained its shape, we say it's turgid and it's well endowed with cytoplasm in it, or cell sap that gives the vacuole its shape. Cell sap as being the contents of the cytoplasm in a cell, and then the term plasmodesmata. Again, structures that allow cells to be connected. So we'll refer to these as we look at the different parts. So let's unpack what the cell theory is. So all living things are made up of cells and are either unicellular or multi. The word unicellular refers to single, so made up of one cell, whereas there are some living organisms that are, many of them are made up of multicellular cells, so organisms that have many cells that make them. And in these we see that the cells have specific functions that ensure that the organism can survive and carry out its normal processes. Cells are the smallest working units of all living things that show the characteristics and properties of life. So in any organism, whether it is a unicellular or multicellular organism, we know that the cell is the smallest component that ensures that this organism can continue with its properties of life. So the smallest living component. All cells come from pre-existing cells through cell division. And so we're going to look at how cells are dividing in our next installment. But it's important that cells are actually divided and come from a pre-existing cell, and we refer to that as division of cells or in the production of more. So we'll continue describing the basic structure of a cell. A cell is a basic structural, functional, and biological unit of all organisms. It is the smallest unit of life. Cells are often called the building blocks of life, essentially because these are what constitute the makeup of organisms. It's important to recognize that in biology, we have the concept of cytology or cell biology, which refers to the study of cells in the focus of how cells work in an organism. So what is a cell? Cells contain the body's hereditary information or material that can make copies of themselves. So cells contain the genetic information that allows for cells to have the same genetic information when they copy from one to the next. Cells have many different parts, each with different functions, and we refer to those as organelles. So smaller little functional units within a cell are called organelles, and these collectively allow for this cell to function as a unit. Some of these parts are called organelles, as I've pointed out, and are specialized structures that perform specific tasks within the cell. And collectively, all of these allow for the organism to be able to carry out its basic survival function. Let's pause and we'll talk about the classification of cells. So we've looked at the cell being a structure that is the simplest unit of life. Within them, there are smaller structures called organelles. And these organelles have specific functions to ensure that collectively that cell as a unit can survive. Based on that, we know that certain structures give a cell specific characteristics and the ability to carry out specific functions. Let's try and understand how we classify cells based on certain structures that are present within the cell. So we'll come across a term now which is referred to as prokaryotes and eukaryotes. And so pro is derived from the word first, and karyotes refer to a term that is defining the karyon, which is the nuclear material. So your prokaryotes are organisms that are made up of cells that lack a cell nucleus or any membrane encased organelles. So these are your simplest cells that have either a strand of DNA but not necessarily a membrane bound organelle. However, when we look at more developed and complex cells, we refer to them as eukaryotes. And these are organisms that have complex organelles that are bound with a membrane, and so they have a defined nucleus which has genetic material within them, and other organelles that have a distinct membrane bounding them. So this is broadly classifying organelles based on them. Let's look at some images of these. So further up on this slide, this would be a prokaryotic cell up here. So here you can see that it's got a simple chromosome, it's got a cell wall, but no distinct membrane-bound structures. It might have simple structures called ribosomes. It will have a bit of cytoplasm, but clearly no mitochondria, no nucleus present. However, if we look at this cell, which is of a eukaryotic organism, we can clearly see defined organelles such as the mitochondria. We can see it's got a defined nucleus, significantly bound with a membrane containing the chromatin network. It's got structures that are complex such as lysosomes and vacuoles. So a much more developed cell having shown specific structures that carry out complex functions. So this would be the classification based on the presence of structures such as nuclei as well as other complex organelles. Again, if we were to compare these two in a diagrammatic illustration, here we see an example of a bacterium cell having a cell wall having some genetic information in the presence of strands of DNA. No significant organelle bound by a membrane in these. However, if we look at a eukaryotic cell, you can see that it shows you a nucleus, it shows you the Golgi apparatus, it has complex structures such as the endoplasmic reticulum. Again, signs of a well-developed organelle development. So the characteristics of a prokaryotic cell would be that they know true nucleus, they have strands of DNA or RNA that are not bound by a membrane. They show few internal structures. An example of this would be bacteria or even some of your viruses. If we compare that to the characteristics of your eukaryotic cells, these have a true membrane-bound nucleus. They have structures that are surrounded by a membrane, and they have complex internal structures such as mitochondria and Golgi apparatus, etc., as we see in this illustration. Cell size. Let's go back to understanding what cell size is. So remember that cells were not clearly visible initially when scientists were curious about trying to unpack that. With the development of complex tools such as magnifying glasses and a microscope, we were able to see further into the details and structure of cells. And that made it much more easier for us to understand the context of what a cell is. Because they are microscopic, it would mean that we cannot see them with the naked eye. We would need to use some magnifying equipment to be able to see these organelles with a better resolution. So cell size. So cells differ in size, and the average size is 10 to 20 micrometers. Now, let me remind you of what a micrometer is. So we have a centimeter, which is further divided up into millimeters. If we take a millimeter and divide that by 1,000, we see that each part contains now 1 micrometer. So take a millimeter on your ruler, divide that into 1,000 parts, and that represents 1 micrometer. And so we can further divide that into a further division, but we will hold on to that as we look at the scale of magnification of different organelles or organisms. Note that most cells are too small to be seen with an unaided eye or without a magnifying equipment. We refer to these cells as being microscopic. So here is a comparison of living organisms and their general size. And so here we have a scale of 1 millimeter, which we will start off with, and then we will work our way up and then further down, decreasing in size. So generally, if we were to look at a fish egg, it's slightly larger than a millimeter or so, and you'll find that a chicken egg could be between 7 to 8 centimeters in length or in size, and we get to the height of a human could vary between 1.7 to 1.8 as an average height of an individual, probably much less. And then you have some giant trees which really are 100 meters in height. So that's kind of easily visible with the naked eye or an unaided eye. However, if we go below organisms or structures that are significantly smaller than a millimeter, we will need to have some magnifying equipment, and in this case, a microscope or a light microscope. So amoeba can be seen with a microscope, and that is between 100 micrometers. So it's again submicroscopic, it's really small, cannot be seen with the unaided eye. A human blood cell is around 7 micrometers in diameter, so that's where we see the red blood cell. If we look at certain the bacterium and their mitochondria, we can see that they're now even smaller, and that's between 2 micrometers in length or width. Viruses measure even smaller, and we get to now a sub-microscopic resolution where we would need an electron microscope to be able to see these. And further down, if we were to compare that size to a hydrogen atom, which would be 0.1 nanometer. So we've gone from micrometers to nanometers. So I'm going to share with you kind of a presentation which shows you the comparison between a human hair, and I'll play this through as we wrap up this session. So here is an illustration showing you the size of the human hair and the scale. It's important that we read off the scale on this axis here, and you can clearly see that the magnification starts on a scale of 1 millimeter and it goes down. If we click on the next, so that's the human hair. Let's look at the size of the human hair in relation to a dust mite, and you will see that now we're looking at probably around 90 micrometers. If we go further down to a pollen grain, and you can see the pollen grain now measures around 1,000 micrometers. If we go further to compare that to a lymphocyte, which is a type of blood cell, we can see that resolution becoming even more smaller. Compare that to a red blood cell. So a red blood cell is much smaller than the lymphocyte. And if we compare that to, again, the yeast that is used in baking, we're going further to a size of a cell that is between 1,500 micrometers. E. coli, which would be bacteria that are rod-shaped bacteria that are found in your intestinal flora, are between, again, 10,000 micrometers in length. Streptococcus, again, a bacteria that we're seeing are much smaller. And then we get to viruses, which are significantly smaller, and these are now in nanometers that can be scaled up into extremely microscopic, where you would need to have an electron microscope to be able to see them. And probably one of the smallest microorganisms would be a rhinovirus, which is measured probably less than 20 nanometers in diameter. So guys, we've looked at the comparative size of organisms. These are cellular organisms that are made up of one single cell, and we notice that they can go from being significantly large as a red blood cell to being significantly small in terms of their size, being in nanometers. And all of these would require highly specialized equipment such as electron microscopes to be able to see them and their definition. Well done, guys, on staying tuned to this session on cells and their structure. We're going to get back and we're going to look at the details of organelles. So have a little stretch break. See you back in a bit. Welcome back, life science learners, to your next installment on cells. So we've looked at cells, we've looked at the structure of cells in terms of their size, and we've looked at classifying them based on the presence of a defined nucleus, making them eukaryotic, or the absence of membrane-bound structures, making them prokaryotic. We also looked at the sizes, and we said that their sizes can differ from being in micrometers to being extremely small in nanometers. Let's try and unpack the structure of organelles and their functions in this segment. So an overview of these cells, and I know that these are having complex structures that are present. So let's identify and look at each one of them separately, and then we'll zone in on each of these organelles and unpack them. So when we look at an animal cell, we can clearly see that there's a significant difference in their shape. Plant cells tend to have a well-defined cell wall, which you will see, which is present in a plant cell, whereas animal cells have a clear, distinct cell membrane. Within an animal cell, you'll find the presence of many mitochondria, and these are structures that are important in cellular respiration. We know that we see them in plant cells and are generally present to also produce energy during respiration. What is unique to a plant cell would be the presence of organelles called the chloroplast, and these are crucial to the process of photosynthesis. They also contain a large vacuole, which is significant to giving the cell its shape and giving it form. We do find vesicles or we do find vacuoles in plants, in animal cells, but they generally are smaller or not present if they are found. What is central to the cell is the nucleus, and this is present in both plant and animal cells, and we will have to look at the structure in much detail as we zone into that organelle. We also know that there are many important structures like the endoplasmic reticulum, which brings the contents of the nucleus in closer proximity with the rest of the cell, and we see that organelle present significantly in plant and animal cells. The presence of the endoplasmic reticulum can be seen dotted with the ribosomes, as you would see here, and we refer to that as rough endoplasmic reticulum, or we see the smooth endoplasmic reticulum, which would be showing the absence of ribosomes, again another organelle. A very important structure, an organelle to look at, is the Golgi apparatus, and these are basically your dispatching units that prepare some of the vesicles with enzymes for protein digestion. So that's a very broad overview of these structures. Let's now focus on each of them with greater detail. So central to a cell is an organelle, which is called the nucleus. I've zoned in on the nucleus and been enlarging that significantly. The nucleus is a membrane-bound structure, a double membrane-bound structure, which is also called the nuclear envelope. It has these nuclear pores or openings which allow for the movement of substances into and out of the nucleus. Remember that the nucleus is the control center, and it has the ability to be able to send out messages to the rest of the organelles within the cell. Central to a nucleus is the nucleolus, which has an important function which we need to unpack in a bit. The nucleus contains a matrix called the nucleoplasm. So that's the liquid part which contains the nuclear material, in this case, which is called chromatin. And this chromatin is essentially threads of DNA that are wrapped together. So that's the overview of the nucleus. Let's try and understand the structure in a bit more detail. So as I mentioned, it's a double-layered membrane. It has pores around it, which allows for the entry and a movement of molecules through them. It has a nucleoli or a nucleolus singular, which are tiny small bodies found in the nucleus. The nucleus has its components suspended in the matrix or the liquid part called the nucleoplasm. Important to the cell is the nucleus in terms of controlling the core functions. So the primary role of the nucleus is to control and regulate the cellular activity of growth and maintaining cell metabolism. So this is controlling the process of cell division as well as maintaining the process of cellular functioning. It also carries genes, which is the chromatin network, that carry the genetic information from one generation of cells to the next. So important in the process of cell division is the genetic information being passed from one cell to the next. The chromosomal DNA and the genetic materials, which are made up of the genetic code for important proteins, which are made up of amino acids. So the sequence of those amino acids that make up proteins are determined by the genetic code in the DNA. So when we talk of protein synthesis, it is the information that comes from the DNA in the form of a genetic code that makes up the specific proteins that are needed in the body. The next organelle that we're going to look at is the mitochondria, often referred to as the powerhouse, and these are found in both your plant cells and your animal cells. It's an oval-shaped structure which is made up of folds of membranes called cristae, and as you see, these are folded membranes on the internal surface. It has an outer membrane and an inner membrane space which is folded. The inner membrane is extended with folds called the cristae, as you would see there. Dotted on the membranes are structures or organelles called the ribosome. The DNA present in the mitochondria is unique, and it's called mitochondrial DNA, which is something that we discuss as an important component of DNA in terms of working out maternal ancestry in grade 12. The DNA is important to ensuring that the DNA codes for specific enzymes or proteins that the mitochondria need. The contents of the mitochondria, the liquid content, is called the matrix. And so that's the overview of the structure of the mitochondria. The size of the mitochondria may vary between 0.5 to 10 micrometers, so it's extremely small. They have two specialized membranes, as I discussed, the outer and the inner membrane, and they have a central mass of a gel matrix present in them, and the folds are known as the cristae. Let's look at the primary function of the mitochondria. Its primary function is to generate energy, and that is through a process called cellular respiration, where the glucose is broken up to produce an energy-rich molecule called ATP, or adenosine triphosphate. So ATP is an energy carrier molecule called adenosine triphosphate. So it's a molecule having three triphosphate molecules. It provides the energy that a cell needs to move, to divide, and to produce secretory products, to contract. In short, they are the powerhouse of the cell. The next organelle that we need to focus on is the endoplasmic reticulum. So the reticulum refers to a maze of membranes that are connected. So this is a structure that connects the nucleus to the rest of the cell. So it brings the contents of the nucleus closer with the rest of the cytoplasm. So the folds on the endoplasmic reticulum are called the cisternae. Dotted on the endoplasmic reticulum are organelles called the ribosomes. So when it is dotted with ribosomes, it's called rough or granular endoplasmic reticulum. When the endoplasmic reticulum has no ribosomes on it, we refer to it as being a smooth ER or endoplasmic reticulum. So that's the structure. So this is a continuation of the folded membranes which the organelle is found in the cytoplasm, and this is an extension, as I mentioned, of the nucleus. And this basically brings and interconnects the nucleus to the contents of the cytoplasm externally. Functions of the endoplasmic reticulum, or ER. It's important in manufacturing, processing, and transporting the proteins for cellular utilization, both in and out of the cell. So this basically allows for the process of transporting the contents in and out of the cell. The endoplasmic reticulum has more than half of the membranous cell contents. Hence, it has a large surface area which is important for chemical reactions to take place. So these folds that you're seeing of membranes basically increases the surface area for more enzyme activity to take place, making this an efficient organelle for cellular function. So it also contains enzymes that basically are important in lipid or fat synthesis, and hence we say that they are an important component in the production of fats that are important to the cell. The next important organelle are ribosomes, and the ribosomes are extremely small, significantly smaller than the endoplasmic reticulum. They're found dotted on the ER. The endoplasmic reticulum is an important organelle that plays an important role in the production of an amino acid chain, or what we refer to as your proteins. In grade 12, we spend a lot of time looking at protein synthesis, where we see this structure called the ribosome, which is essentially made up of two units. It's called a small subunit and a large subunit. And this is a structure that actually reads the mRNA and produces a chain of amino acids, which essentially now becomes a protein molecule. So the ribosomes are made up of ribosomal proteins and RNA. Each ribosome is made up of two subunits. It has a large subunit and a smaller subunit, and these collectively carry out the function of protein synthesis. We're going to race on to the Golgi apparatus, and here is an illustration of a two-dimensional image as well as a three-dimensional image of how these would appear in the cytoplasm. So these are folded membranous structures which produce vesicles that bud off at the ends, and these vesicles contain important enzymes or secretions that are important in the functioning of the cell. So again, these cisternae are folds of membranes that break off, which have an internal lumen, so they're hollow but contain a secretion on the inside that bud off and form vesicles, which are smaller little vacuoles. So these are membrane-bound cell organelles. They're found in the cytoplasm of eukaryotic cells, again much more complex cells, and they're often very close to the endoplasmic reticulum and near the nucleus. So this connects the nucleus and the endoplasmic reticulum, which coordinate the packaging and production of important enzymes and proteins. The Golgi bodies are supported together by the cytoplasmic microtubules, which keep them held in position in the protein matrix. It is made up of a flattened stack of pouches known as the cisternae. The functions of these is that they transport and modify and they pack proteins and lipids into the Golgi vesicles, which are those budding structures that we see here. And so they contain important proteins and often enzymes that have a specific target site which they move to. Animal cells may contain one or more Golgi bodies, while plant cells have a few hundreds of these. And that talks to the functioning of these structures in these organelles. As we change onto plant cells, we know that plant cells are characterized by the chloroplast. And the chloroplast is an organelle that contains the necessary structures that are responsible for photosynthesis. Let's try and unpack what the structure of a chloroplast is. So it's a membrane-bound structure, again a double membrane-bound structure, which contains an outer membrane and an inner membrane. Within it, it has these stacks of coins, which I refer to as thylakoids. So these are coin-like structures or membrane-bound structures that are stacked up to form grana or granum, and these basically are structures that contain the pigment called chlorophyll, and it is within these structures that the process of photosynthesis takes place and the production of a complex carbohydrate called starch. Okay, these are connected to each other by intergranum lamellae. What is unique to the chloroplast is that it has its own DNA that produces or codes for information that produces important enzymes and proteins. The structure of the chloroplast, we said, are found in plant cells only and in algal cells. They are oval shaped and they carry out important processes. Let's look at that. They have two surface membranes, an outer and an inner membrane, as I discussed, and the inner membrane is known as the thylakoid, which has two membranes that are folded together. The outer membrane forms the external lining of the chloroplast, while the inner membrane forms below the outer layer. The function of the plant cell in plant cells of the chloroplast is that it is a site for protein synthesis, for food synthesis. When I mean food, I mean glucose, and that is the material that is produced by the cell. So it's a carbohydrate, and that process is known as photosynthesis. And so they contain a pigment called chlorophyll, which is crucial to the absorption of light and conversion of that light energy into chemical energy. So guys, that's a wrap for this section. We've looked at some of the organelles. The most important ones were discussed. So we looked at the nucleus, we've looked at the mitochondria, we've looked at the chloroplast, and we're going to look at some of the other organelles in the next segment. So have a little break, stretch a bit, grab some water, and see you in a bit. Welcome back, life science learners. In this segment, we're going to wrap up the structure of the cells. We're going to focus on some applications of our understanding of the different organelles, and let's try and look at how these relate to understanding the context of a cell and its functions. Before we get onto that, we need to discuss a very important structure, which is the cell wall, which is unique to plant cells, and often we use this as a differentiating factor between a plant cell and an animal cell. Let's look at the structure of a plant cell wall. So I've got a very complex diagram that illustrates the structure in much detail. The detail that I've shown to you is not important for you to remember, but it's important for you to recognize and understand that this cell wall that we talk about is a complex structure that has a very significant function in the plant cell. So it is made up of different layers. So we have a middle layer called the primary cell wall, which is enclosed by an outer middle lamella and an inner plasma membrane. And these are what connect to an adjacent cell. So imagine having a sandwich, and this is essentially what the filling is, and on either sides you have the outer and the inner slices, and that is what connects this membrane to the next cell. Within them you find some complex carbohydrates called pectin, you've got cellulose microfibrils, and you've got soluble proteins, which make the movement of substances through this much more effective. So the structure of the plant cell wall is that it is a specialized matrix that covers the surface of the plant cell. So essentially it is the skin of the cell. Every plant cell has a cell wall layer, which is a major distinguishing factor between a plant cell and an animal cell. The cell wall is made up of two layers, a middle lamella and a primary cell wall, and sometimes a secondary cell wall, which we will see in much more complex plant cells. The middle lamella acts as the strengthening layer between the primary walls and the neighboring cells. So when you look at this, you can see the middle lamella contains these complex pectin and cellulose microfibrils that essentially give the strength to the cell wall. So the function of the plant cell is that it provides the cell with mechanical protection and shielding the cell from chemically harsh environments provided by the secondary cell wall. So it's important in that it provides protection and shielding in terms of from the external environment. The plant cell wall is a semi-permeable membrane. Essentially, semi-permeable meaning it's not impermeable. It allows for some substances to pass through, and hence semi-permeable. Okay, so it allows movement in and out and the circulation of materials such as water, molecular nutrients, and minerals through the membrane. It also provides support in terms of a rigid building block to stabilize the plant to produce some of its structures. For example, the stem and leaves of the plant. So when we discuss the structure of a cell, especially a plant cell, we know that it lacks an exoskeleton, but its strength comes from the middle lamellae, where it has these cellulose fibers, the pectin, which gives it structural shape and form. Right, so it's time that we now compare the differences between a plant cell and an animal cell. This is often a question that comes up as a comparison question so that it will test the understanding between the two. So most plant cells contain plastids, whereas animal cells do not contain these. And these are organelles that allow for the plant to be able to photosynthesize and give them color. So the various colors that you see in the leaves and in the color of the fruit comes from the different types of plastids. A plant cell is surrounded by a cell wall and a cell membrane, whereas an animal cell has only a cell membrane surrounding it. Plant cells usually may have one or a large vacuole present that gives the cell its shape and structure, whereas animal cells may have none or even a few small specialized vacuoles may be present. Plant cells generally have a regular, rigid shape, whereas animal cells could have irregular and diverse shapes depending on where their structure and form is. Let's look at the following question and try and apply our understanding to this. Study the diagram below and answer the questions that follow. So here's a diagram that shows various parts that are labeled or require labels. So as a quick overview, in a test or an exam, it's good to have an overview of the diagram, look at the diagram and try and establish the different labels that have been provided. Then read the questions to try and get a better understanding. I clearly see a well-developed nucleus. I see the presence of mitochondria. I notice the Golgi apparatus. I also see a small vacuole. I can see the remnants of what we call the centrioles that are important in cell division. And we can see some other organelles like the smooth endoplasmic reticulum. So that's an overview of the different structures. I'm going to now focus on the next part of the question. Identify whether the cell below is an animal or plant cell. Provide a reason for your answer. If we go back to that diagram, let's look at it. So when I look at this diagram, I can clearly see it's a membrane-bound organelle or structure having a single membrane, so that's certainly the cell membrane. So there's no cell wall present, so I can clear a defined cell membrane with the absence of a cell wall. I do see a vacuole which is significantly small, which is unique to an animal cell. If this was a plant cell, it would have had a significantly larger vacuole. Okay, so we can clearly see two distinguishing characteristics that give us pointers to why this is an animal cell. Okay, right. So provide labels for the following parts that are labeled B, C, D, E, F, L, and J. So I'm going to point to those structures in the next slide and try and label them. So we've got to go to B. So B, if we look at B, B is pointing specifically to structures within the nucleus. And that's essentially your chromatin material or your DNA. So that's your chromatin network. So that's the chromatin material. That's B. C points to the structure here, which is the nucleolus. D points to the matrix or the liquid part of the nucleus called the nucleoplasm. E points to, on the surface, on the outside, close to the nucleus, is the endoplasmic reticulum. The question is this a smooth or rough ER? Clearly, no presence of ribosomes on the surface, so this is a smooth ER, meaning that it shows the absence of the ribosomes on it. We also see that this structure here, which is F and G, points to the Golgi apparatus. So that's the Golgi apparatus with the vesicle present in there. Okay, so if we go back to that, we can clearly see structure B is the chromatin network, which we got correct. C was the nucleolus. E was the contents of the nucleus called the nucleoplasm, and we identified structure E as the smooth ER. F was the Golgi apparatus, and if I go back to structure L was the vacuole, and structure I, if I'm correct, was the okay, so we just got to check that structure L was the vacuole, and structure J, if we go back to J, J is here, and J points to this structure, which is the endoplasmic reticulum. But again, these are dotted with ribosomes, so that's the rough endoplasmic reticulum. Cool. Describe the function of part labeled num L. So if we go back to the diagram, part labeled L was the mitochondria, and if we recollect, the function of the mitochondria is that it is producing energy in the form of ATP during cellular respiration. So that is for cellular respiration and the production of energy. Cool. Let's move on to some more questions. Describe the function of part label 1, which we've put down. It's basically the powerhouse that produces energy through respiration. And to wrap up, discuss how the part labeled I, which is the mitochondria, is structurally suited for its function. Guys, as we wrap this section up, that's an important question that kind of leads to the way teachers often question the structure of an organelle. So for each organelle, you should know the structure, you should know its function, and you should know how it's adapted for its specific function. With the example of the mitochondria, we know that it is membrane bound. It's got these folds called the cristae, which increase the surface area for greater enzyme functioning. So guys, it's been a rapid, quick third session where we've looked at some important functions of the different structures. We've compared plant and animal cells. I hope you've enjoyed the session. Go back and review these notes and try and understand and recollect how these structures are suited for their functions. Thank you for staying tuned. Keep well. See you soon. Enjoy.